Dear students, in our previous session, we completed the discussion over Mendel's monohybrid cross. If you are knowing about the entire concept of the monohybrid cross or the inheritance of one gene, the today's discussion of Mendel's dihybrid cross, or it can be called inheritance of two genes, become very easy for you. Before starting with this, let us just recall the definition of inheritance of one gene. One gene means Mendel's monohybrid cross. How we will define that? It is the cross involving two true breeding parents. Parents should be always what? True breeding. What do you mean by true breeding? They should be genetically pure. ಅಂದರೆ ಹೋಮೋಸೈಗಸ್ ಕಂಡೀಷನ್ ಎಲ್ಲ ಇರಬೇಕು ಅಂತ ಅರ್ಥ ಅಲ್ವಾ. ಕ್ರಾಸಿಂಗ್ ದ ಟು ಟ್ರೂ ಬ್ರೀಡಿಂಗ್ ಪೇರೆಂಟ್ಸ್ ವಿಚ್ ಡಿಫರ್ ಇನ್ ಒನ್ ಕ್ಯಾರೆಕ್ಟರ್ ಈಸ್ ಕಾಲ್ಡ್ ಇನ್ಹೆರಿಟೆನ್ಸ್ ಆಫ್ ಒನ್ ಜೀನ್. But today I am going to discuss inheritance of two genes. How to define? Very simple. It is the cross involving two true breeding parents which differ in how many character? Two character. Two true breeding parents which differ in two character is called inheritance of two gene or the dihybrid cross. In monohybrid cross, he selected only one character. He focused on that only. Which is that? It is height. You know that height was available in two traits. One is tall, another one is dwarf. True breeding tall plant, true breeding dwarf plant. He crossed. He got F1 generation. The F1 progeny was later self-pollinated to get which one? F2 generation, right? Here, as it is a dihybrid cross, or we are focusing on how two genes or the two characters get transmitted from one generation to another. So, in case of dihybrid cross, Mendel selected two characters. What are they? In that plant, one character is seed shape and another one is seed color. Okay, at a time throughout the experiment he focused on two characters. One is seed shape, and you already know that seed shape is available in two traits. What are they? One is round seed and another one is wrinkled seed, capital R, small r. Whereas seed color is available in two traits. They are, one is yellow, represented by the gene or the allele capital Y because it is dominant. Another one is what? Green, represented by small y. So throughout the experiment, Mendel focused on two characters with its contrasting traits: seed shape, seed color, round, wrinkled, yellow, green. Now, how he performed the experiment? Very simple. First, he took two true breeding parents. Parents number is always 2, and you know the parents should be always what? At the beginning of the experiment, what you are taking them? They should be in the homozygous condition. They should be true breeding. He crossed round seeded yellow colored pea plant, yellow colored true breeding pea plant with which one? Wrinkle seeded green color pea plant, the true breeding pea plant. Remember that both the plants are true breeding, they are homozygous, and hence what is the genotype? Okay, the genotype is round is governed by what? Capital R, yellow by capital Y. Dihybrid cross ಎರಡು ಕ್ಯಾರೆಕ್ಟರ್ಗಳ ಬಗ್ಗೆ ನೀವು ಫೋಕಸ್ ಮಾಡೋದ್ರಿಂದ ದ ಜೀನೋಟೈಪ್ ಆಫ್ ದಿಸ್ ಪ್ಲಾಂಟ್ ಈಸ್ ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಕ್ಯಾಪಿಟಲ್ ವೈ ಡಿಪ್ಲಾಯ್ಡ್, ರೈಟ್? ಹಿಯರ್ ಸ್ಮಾಲ್ ಆರ್ ಸ್ಮಾಲ್ y. Both the plants are ready. First step in the reproduction is formation of what? Gametes. Now the first plant formed the gametes and all gametes had capital R capital Y. Gametally capital R capital Y. In this case all gametes had small r small y. Gametes are ready. You will perform emasculation, dusting, right? And thereby you conduct self-pollination or cross-pollination. Cross-pollination. ಈ ಎರಡು ಪ್ಲಾಂಟ್ಗಳ ರೈಟ್ ಮೇಲ್ ಗ್ಯಾಮೆಟ್ ಫೀಮೇಲ್ ಗ್ಯಾಮೆಟ್ ಅನ್ನ ಏನು ಮಾಡ್ತಾ ಇದ್ದೀರಿ? ಕ್ರಾಸ್ ಪಾಲಿನೇಟ್ ಮಾಡ್ತಾ ಇದ್ದೀರಿ. ಬಿಕಾಸ್ ಆಫ್ ವಿಚ್ ಯು ಆರ್ ಎಂಡಿಂಗ್ ಅಪ್ ವಿತ್ ದೇರ್ which generation? First generation. I call it F1 generation. So when they are cross pollinated, the progeny formed in the F1 generation, a first generation plant ಏನು ಫಾರ್ಮೇಷನ್ ಆಗ್ತಾ ಇದೆ, that had a genotype capital R small r capital Y small y. The mistake that you do is capital R capital Y small r small y ಒಟ್ಟಿಗೆ ಬರಿತೀರಿ. No, it is not like that. How you have to write? Capital R small r, right? Capital Y small y. Why you are writing capital R small r together? Why not capital R capital Y ಅಲ್ವಾ? capital R capital Y ಬರ್ದು ಆಮೇಲೆ ಸ್ಮಾಲ್ ಆರ್ ಸ್ಮಾಲ್ ವೈ ಬರಿಬೋದಲ್ವಾ? ಬಿಕಾಸ್ ಯು ಆಲ್ರೆಡಿ ನೋ ದ್ಯಾಟ್, ರೈಟ್? ದ alleles of a gene are present at same locus on a homologous chromosome. The alleles of a gene are present at same locus. That is the reason why you are writing capital R small r capital Y small y. Is that clear? Be careful. It is homozygous or heterozygous? The F1 plant is heterozygous, right? That's why it is called F1 hybrid. It is called what? F1 hybrid. What is the phenotype? This is the genotype, genetic combination of the plant. Can you guess what is the phenotype? Capital R is present, hence it is what? Round. Capital Y is there, hence the seed color is what? Round shape. Yellow color seeded plant was born in the F1 generation. Again, Mendel want to see what happened to another parent character. When he crossed these two plants, in the F1 generation, he got a baby plant which had only one parental character. What happened to another parental character? Right? To check that, and you know, in this case, why the plant had round seeds? Because capital R is dominant, small r is recessive, right? According to law of dominance, capital Y is dominant, small y is recessive. Now, for the next 5 minutes, try to understand. He want to know what happened to another parental character. To check that, he thought of obtaining its next generation. Which is its next generation? That can be called F2 generation. How you will get F2 generation? He thought of doing what? Self-pollination. F1 into F1. Self-pollination. Is that clear? Now, can you guess? You already know the genotype of the F1 plant. What is the genotype? Capital R small r capital Y small y, capital R small r, capital Y small y. A bisexual flower which will be what? Self-pollinated. Now, first step during the pollination and fertilization is what? Formation of gametes. Formation of the gametes. F1 plant formed the gametes. Question is, all gametes are of same type? No. Here, few gametes carried, see, very important, capital R, capital Y. Few gametes had capital R, small y. Few had small r, capital Y, small r, small y. Let me write once again. Right? Few gametes formed here are capital R, small r. No, not small r, capital R, capital Y. Right? Here, the mistake that you do is you will write capital R, small r. No, chromosomes will segregate, they will separate, right? Law of segregation. Capital R, small y. Next, small r, capital Y, then small r, small y. Is that clear? Any confusion? Please don't do the mistake of writing capital R, capital Y, small y. Wrong. ಹಾಗೆ ಬರಿಬಾರ್ದು. ಯಾಕೆ ಅಂತ ಹೇಳಿದ್ರೆ ಡ್ಯೂರಿಂಗ್ ಗ್ಯಾಮೆಟ್ ಫಾರ್ಮೇಷನ್ ಈ ಪ್ಲಾಂಟ್ ಇಂದ ಅಲ್ವಾ ಕ್ರೋಮೋಸೋಮ್ಸ್ ವಿಲ್ ಸೆಗ್ರಿಗೇಟ್ ಅಂತ ಹೇಳಿ ಯಾರು ಹೇಳಿದ್ದಾರೆ? ಮೆಂಡಲ್ ಇನ್ ಈಸ್ ಲಾ ಕಾಲ್ಡ್ ಲಾ ಆಫ್ ಸೆಗ್ರಿಗೇಷನ್. ಈ ಒಂದು ಕ್ರೋಮೋಸೋಮ್ ರೈಟ್ capital R ಒಂದು ಗ್ಯಾಮೆಟಿಗೆ ಬಂದ್ರೆ okay, the another gamete is going to receive small r. Say for example, here one is going to get capital Y, another going to get which one? Small y. Likewise, different combination can be possible. So you have to be very careful. The gametes formed by the F1 plant are capital R, capital Y, capital R, small y, small r, right? Capital Y, small r, small y. The F1 plant formed how many different gametes? 4 different gametes. Now, when the male gamete and female gamete of a bisexual flower, when they are self-pollinated, when the plant is self-pollinated, what are the possible combination of genotype and phenotype of the progeny? ಇವರಿಬ್ಬರನ್ನ self pollination ಮಾಡಿದವಾಗ right, what is the combination of the genotype and phenotype of the progeny? ಇವರಿಗೆ right, ಇವರ progeny ಏನು ಫಾರ್ಮೇಷನ್ ಆಗ್ತಾ ಇದೆ? progeny ಅಂದ್ರೆ ಯಾವ ಜನರೇಷನ್? F2 generation. ಅವರ genotype phenotype ಅನ್ನ ಬರೆಯೋದಕ್ಕೋಸ್ಕರ I will once again going to take the help of what? Checkerboard. Can you recall along with me? Who proposed this? Reginald C. Punnett. See, in the monohybrid cross, a plant formed 2 different types of gametes, right? One is capital T, another gamete had small t. 2 gamete when it is there, 3 rows, 3 columns. Here, 4 different gametes, hence, right? I'm sorry, one more is missing here. Let me write it here. 5 rows, 5 columns. Clear? Okay. When 4 gametes are there, 5 rows, 5 what? Columns. Now, here, according to Reginald C. Punnett, the top row as well as on the left hand side should be used for writing what? Gametes. The male gamete and female gamete. Now consider these 4 as the male gamete. What are they? Capital R, capital Y, easy. Capital R, small y. Now small r, capital Y, small r, small y. Correct, no? Write down once again. Capital R, capital Y. Follow our order, don't change. Capital R, small y. Now small r, capital Y, small r, small y. Gametes are ready. Now let us see their combination. Please write the combination along with me without wasting the time. Okay, now suppose if this female gamete is fertilized by this male gamete, the genotype of the progeny will be capital R, capital Y, capital Y. As you write the genotype, immediately you have to write the phenotype of it. What is the phenotype? Phenotype is round yellow. Okay, likewise, instead of this, if the female gamete is fertilized by male gamete having capital R small y, then the genotype of the progeny will be capital R, capital R, capital Y, small y. Once again, what is the phenotype? It is round yellow. Now, if it is the case, hope you can understand. Again and again, need not to repeat. If this gamete is fertilized by a male gamete having small r, capital Y, then capital R, small r, capital Y, capital Y. Then also what? Round. Why round? Because capital R is dominant. Another one is yellow. In this case, it will be capital R, small r, capital Y, small y. Once again, it is what? The progeny formed will be round yellow. Can you continue along with me the same thing? It is very simple, dear students. Please finish it off along with me. Capital R, capital Y, small y, round yellow. The mistake that you usually do is you will first keep on writing the genotype. Later you will get confused here and there. As you write genotype, have a practice of writing which one also? Phenotype also. Genotype, phenotype, both you have to write. Okay. Capital R, small y, it will be what? Round and green. Right now, capital R, round, small y is present. Small y responsible for green. There are no, what is that? Dominant allele is present for the seed color. Capital Y is not there and hence it becomes green. What about in this case? Capital R, capital Y, small y. It is round yellow. Okay, now again here capital R, small y, round what? Green, round green. In this case, capital R, small r, capital Y, round yellow. Again, capital R, small r, capital Y, small y, it is also what? Round yellow. In this case, hope you are practicing along with me. It is very simple. Look at here, you have to be very careful. Small r, capital Y. Here both are recessive alleles. Hence, what is the seed shape? It is wrinkled, and here there is a dominant allele, hence the seed color is what? Yellow, wrinkled yellow. What about here in this case? Small r, capital Y, small y. Once again, it is wrinkled yellow. Now the last one, let us complete this. Capital R, capital Y, small y. Okay, here dominant alleles are there, hence seed shape is round, seed color is what? Yellow. What about here? Capital R, small y. It is round, small y. There are no dominant allele for seed color, hence it becomes what? Green. In the absence of dominant allele, it gets the recessive character. It is green. Last two, what is that? Small r, capital Y, small y. It is both are recessive allele for seed shape. Hence it is wrinkled, right? Yellow. Last one, if you look at this, small r, small y. It is here both the pairs of alleles are in the recessive state. For the first time, it is wrinkled green. Okay, so this is how in the F2 generation, when you self-pollinate the F1, possibility of 4 different types of gametes from the F1 plant, right? 4 different types of gametes from the F1 plant. How many possible combinations of progeny? It is 16, right? 16 variety of combination of progeny can be totally when we see. Now, in this, if you look at the phenotype round yellow, let me write here, how many of them are round yellow in this case? How many of them are round yellow? I will use the red ink, okay, and it is even your responsibility in the board's exam. You have to check it properly. Here, 1, 2, 3, 4, okay, 5, 6, then 7, 8, one more is here, right? 9. Totally, how many are there? Out of, right, 16, 9 are round yellow. So round yellow is one of the phenotype, one of the character that I can see in the F2 generation that resembles one of the parent. Round yellow ಅನ್ನು resemble ಮಾಡ್ತಾ ಇದೆ. ಇದನ್ನ ಬಿಟ್ಟು ಮತ್ತೆ ಯಾವುದಿದೆ ಬೇರೆ? Can you check it here? Give me a minute, dear students. Let me write it like this. Round yellow. How many? They are 9. Apart from that, I can see here round green. One more phenotype that I am going to see here is what? Round green. How many of them are round green? 1, okay, 2, then is there, yeah, here is one more. How many are there? 3. So 3 of the progenies are round green. Apart from that, I am going to see here one more phenotype, that is wrinkled yellow. How many of them are wrinkled yellow? Here is the first one, second one, here is one more. Once again it is what? 3. And another phenotype that I am going to see here, what is that? Wrinkled green. How many? Only 1. So how many different phenotypes we can expect or we can see in the F2 generation? 4 different types of phenotype. What are they? Round yellow, round green, wrinkled yellow, wrinkled green. 4 different types of phenotypes are observed in the ratio 9:3:3:1. So my dear students, what is the Mendel's monohybrid cross phenotypic ratio? I repeat, Mendel's monohybrid cross phenotypic ratio is 3:1. What is the Mendel's dihybrid cross phenotypic ratio? What is your answer? The same that you learnt in the 10th class, 9:3:3:1. Don't forget to write the phenotype. What does the 9 indicates? It is round yellow, round green. This is compulsory that you need to write. Along with the ratio, you have to write what phenotype that particular digit indicates. Is that clear? So this is the phenotypic ratio of Mendel's what? Dihybrid cross. Okay. Now, I just want to repeat this experiment. Try to focus here. As he is checking the inheritance of 2 genes, he selected how many different character? 2 character. One is a seed shape, another one is a seed color. They were available in the contrasting traits. He crossed the true breeding plants. Exam ಗೆ correct ಆಗಿ ಓದಿ ಹೋಗದೇ ಇದ್ರೆ, I have seen such students. That's why I am telling you. ಓಕೆ, ಕರೆಕ್ಟಾಗಿ ಪ್ರಿಪೇರ್ ಆಗಿ ಓದದೆ ಹೋಗಿದ್ರೆ ಏನಾಗ್ಬೋದು, ನಿಮ್ಮ ಏನು ಬರಿತೀರಿ ಅಂತ ಕೇಳಿದರೆ ರೌಂಡ್ ವ್ರಿಂಕಲ್ಡ್ ಗ್ರೀನ್ ಯೆಲ್ಲೋ ಅಂತ ಬರೆದು ಬರ್ತೀರಿ. ಪರ್ಫೆಕ್ಟ್ ಆಗಿ ಓದಿ. ಡೋಂಟ್ ಜಸ್ಟ್ ಸ್ಕ್ಯಾನ್ ಆ್ಯಂಡ್ ಗೋ ಟು ದ ಎಕ್ಸಾಮ್. Clear? Okay, it is not that easy. Practice is required. Round seed, yellow fret colored seeds, pea plant is crossed with this one. Both the plants are true breeding, so hence they are in the homozygous condition. Gametes are formed when they are cross pollinated. The F1 plant was a hybrid with the genotype capital R small r, capital Y small y. Okay, now to see what happened to another parent character, only one parental character was observed in the F1. What happened to another parental character? He thought of obtaining with generation, F2. How he got F2? By self-pollination. And it is very important to write the gametes formed by the F1 plant. Practice this. How many different types of gametes are going to form? 4 different types of gametes, right? Now, with the help of checkerboard, or that is called Punnett square proposed by Reginald C. Punnett, when you completed this, you come to know that 16 progenies were obtained. Whether all 16 were different type? No. The 16 progenies which we got in which generation? F2 generation, they were 4 different type. Mendel thought of obtaining F2 generation because in the F1 he got only one parental character. He want to know what happened to another. In the F2, along with the parental character round yellow, he also got which one? Wrinkled green. Both the parental characters he got in the F2 generation. Along with that, he got 2 more varieties of progenies. ಪೇರೆಂಟಲ್ ಕ್ಯಾರೆಕ್ಟರ್ ಒಟ್ಟಿಗೆ ಅವನಿಗೆ ಇನ್ನೆರಡು ರೀತಿಯ ಪ್ರೊಜೆನಿಗಳು ಸಿಕ್ಕಿವೆ. ದೇ ವರ್ ನಾಟ್ ಎಕ್ಸಾಕ್ಟ್ಲಿ ಸೇಮ್ ಆಸ್ ಪೇರೆಂಟಲ್ ಟೈಪ್. ಪೇರೆಂಟಲ್ ಅಂತ ಹೇಳಿದ ತಕ್ಷಣ ನಿಮ್ಮ ಸೆರೆಬ್ರಮ್ ಅಲ್ಲಿ ಸ್ಟ್ರೈಕ್ ಆಗುವಂಥದ್ದು ರೌಂಡ್ ಯೆಲ್ಲೋ, wrinkled green. ಆದರೆ ಅವೆರಡರ ಒಟ್ಟಿಗೆ ರೈಟ್ ಹಿ ಗಾಟ್ 2 more progenies which were not exactly same as parent, and hence how they were round green, wrinkled yellow. They were a mixture. Hence he called these two as what? Recombinants. Is that clear? So 9 and 1, what is there? These two are parental type, gotirli. Whereas these two are not resembling the parents, and hence I can call these two as what? Recombinant type. Anyhow, whenever we speak about dihybrid cross phenotypic ratio, your answer is 9:3:3:1, right? Now, in this only, can you tell what is the parental type is to recombinant type ratio? What is that? How much it is? Can you tell me? The parental is to recombinant type ratio. It is the parental type are 9 plus 1. It is how much? 10. 3 plus 3 members in this are not resembling parents, and I call them as what? Recombinant type. It is, I'm sorry, it is 3 plus 3, 6. So, dear students, what is the parental is to recombinant ratio? It is 10:6, which can be written as how much? 5:3. Very important. You can expect, write the dihybrid cross phenotypic ratio. This is your 1 mark answer. If they ask you, in dihybrid cross or inheritance of 2 gene, experiment performed by Mendel, what is the parental is to recombinant type ratio? Then your answer is what? 5:3. Okay, 10:6, which can be simplified and written as 5:3. Is that clear? So this is all about the Mendel's what? Dihybrid cross or the inheritance of 2 gene. One of the highly expected question. I will read out the notes that you are supposed to write in the exam. You can have a points of it. Okay. First you have to start with the What is that definition? It is the cross involving two true breeding parents which differ in two character. Next line. Mendel selected two characters. Two characters. Next line, write the same thing. Seed shape, show like this, round wrinkled, capital R, small r. Next line, seed color, yellow, green, capital Y, small y. Next line, write down. Mendel crossed true breeding, round yellow seeded pea plant, having genotype capital R, capital Y, with wrinkle green seeded pea plant, or you can write true breeding wrinkle green seeded pea plant having genotype small r, small y. Next line, along with quality, quantity is also important, right? Neatagi bariri adanna. Next line, in the F1 generation, he obtained a round yellow seeded pea plant having the genotype. In the F1 generation, he obtained round yellow seeded pea plant having genotype capital R, capital Y, small y. Full stop. Continue the line. Here, this is a F1 hybrid. It is a F1 hybrid. Write in your own words. Very simple. Next line. F1 plants are self-pollinated to get F2 generation. Self-pollinated to get F2 generation. Okay. Next line. In F2 generation, he obtained 4 different types of plants. They are, next line, number 1, round yellow, number 2, round green, number 3, wrinkled yellow, number 4, wrinkled green. Next line, the above plants appeared in 9:3:3:1 ratio. Above plants appeared in 9:3:3:1 ratio. Okay, this much. Once you complete the flow chart, entire thing, you are going to get 2.5 mark. And here, please check once you write, you count and confirm whether everything is right or wrong. Otherwise, in the exam, in the board's exam, okay, in that short period of time, what may happen? Sometime you may commit mistake in between. Examiner, evaluator will check every point. So very important. So when you write the ratio, give few seconds of time and quickly check the phenotypes, genotypes. Okay, then you write this. You are going to get 2.5 mark if you write up to here. After that, write the explanation that I have just now dictated. So this is all about Mendel's dihybrid cross. Based on this, he proposed a law. Based on this observation, he proposed a law, and it is called law of independent assortment. About this, I will discuss in my next session.